recording.
Yeah. Yeah. Yeah.
No, I'm talking to No, I'm talking to How bad is it mess up?
Triglyceride?
Can I help you with that?
Yes.
Can I go to the bathroom?
Yes.
No.
Thank you. I feel.
No. All right, go.
I wish it would load. It's giving me stress.
Oh my god, I forgot my AirPods in my pocket got entire.
Alright, can you guys sit down and get settled? We're gonna do lipids today, but I want to talk about your lab, like write-ups first. I have it. Where's your lab? Okay. All right. Shh. So did you guys all? So I'm gonna give you an extra day to do the one that I gave you last night because a lot of these like weren't complete. They weren't like totally. So the reason I wrote out all these things is because I want you to include every single thing. So you should be like checking off every thing here, right? A lot of you guys so it says describe in detail the proper technique for using a micropipette, right, to measure a sample and then how to load it into the wells, right? So it says when to release the button. So I wanted to see that you press the button before you put it into the dye, and then you release it when it's in the dye, right? You press it to the first stop, and and it says which finger to use, right? Because some people had issues like with which finger to use. So you should be saying with your thumb, right? Where to hold the sample when measuring it. Where should you be holding the sample? At eye level, right? But a lot of you guys didn't include those things. You just said press, get sample, get rid of sample, put sample into wells. It was really not clear enough, right? So the reason I'm asking you to do this is because a lot of you guys, when you came up there, even after being drilled and after watching the demonstration, you still didn't do it right? Some of you guys were releasing the thing too soon while it was in the well, and then what was happening? You were kind of drawing up some of the solution back, right? You don't want to do that. Some of you guys were sticking the pipettes too much into the gel, and then you were trying to press the button, and then what happened? You pushed really hard, and then there was this kind of burst, right? That means if you saw this kind of burst, this cloud of dye, that meant that you were in the gel, and you were the thing was clogged because you were stabbing the thing into the gel, right? So that's why I wrote other considerations or precautions to take when loading the sample. I wanted to see that you were opening the sample and holding it in that you were opening the sample and holding it in that other hand, right? And that you were never putting it down. So just a lot of you guys it wasn't that thorough, right? I mean, I know that you know how to do it, but I want to see that you can actually follow the instructions and show that you're pressing it down even as you remove it from the gel, and then you release once it is out, right? And then the second stop, all these little details. So I want to make sure that that's happening. A lot of you guys also, for number 11, it says, in your own words, briefly summarize the purpose of this technique, right? A lot of you guys, I just got a chart or a table, right? I wanted a summary. So I didn't need you to tell me again what you do with the pipette, but what would a good summary of what we did in the gel electrophoresis lab be? Lita, do you want to?
What we were trying to do was trying to figure out which dyes, some of them, if there were colors, positive charge, negative charge, and how big they were.
Good. So the purpose was to determine the charges, the relative charges of these dyes, and then the relative lengths of the molecules within the dyes, right? So that was the purpose. And then what was the procedure? You load dyes into the electrophoresis apparatus. That's all I needed you to say. Then you put what? You turn on an electric current, right? So that the negative electrode would attract the molecules of the respective opposite charges, right? So I wanted to see something like that, but a lot of you guys just didn't put anything in that part, right? And then the other thing is, so the question about what can you conclude about the dyes that produced one band? A lot of you guys said the dyes were the same size, or they are only one size. What's wrong with that? Are the dyes themselves the same size? No, it's the molecules within the dyes that are the same size or of different sizes, right? So that's like, just be more precise with your language when you're working on these kind of things. And if I give you things where it says include when to blah, 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 you should be checking those off to make sure you're including those things, because that's like, exactly what I'm telling you what I want, right? And I think that was about it for this one. So I didn't, I'm not going to take points off of this one, but I did sort of show you, I'm going to show you, I didn't get through all of them either because, there are a lot of them and a lot of them weren't like the way I wanted, so I was like writing a lot on them. So I'll give you guys these back tomorrow, but I'm going to give you one extra day to do your gel column ones, just to make sure you're doing it like thoroughly. Also, so you guys all shared with me your graphs, right? So did you all do graphs or did you only send me a table?
I did both.
Yeah. A lot of you guys did give me a graph, but a lot of people just sent me a table of values, right? It says a graph and it says a table and a graph, right? How should I make this graph? What do you think would be the best way to do it, George?
I did a line graph, and I had each sample had its own thing.
So which one was on which axis?
So the samples was on the x-axis.
All right. So it was sample what? Like one, so A, B, C, like this?
Oh, wow, no, 1, 2, 3, 4, 5.
All right, 1, 2, 3. Okay. Oh, right. Sorry, I'm getting confused with the dial. Okay, 1, 2, 3, 4 through 8, right?
Yeah.
And then
on the y-axis, I had the amounts going from
the absorbent.
0 to 0.2.
So 0.0 to 0.2, right? And then you had it something like, so it looked something like that?
Yeah, there was 2 lines.
Do you know what this line is?
And then there was a key in the top right for each line. So the orange, I think it's orange and blue. And the orange one was for
orange and yellow? Yellow. The
No,
dye?
I'm saying for the graph,
Oh, okay.
there was a line. The orange line was for a 420 wavelength.
Okay.
And the blue line was for 620.
Okay, so you showed. Oh, so one of them was for, and then so you did each sample for each.
Yeah.
Okay, and
And
that's
then there was another line that was
that
for absorption.
presumably would have been opposite of that one, right? Because the greater absorbency of the ones,
Yeah.
so it would maybe do something like this, right?
Yeah.
Okay. All right. So line graph, that was one way of doing it. Did anyone else do a different way?
I did a bar graph with the values.
All right, I think a bar graph also works well. So if you do a bar graph, you would do what? What would you be on the
I
x-axis?
did the same thing.
The samples, right? So the samples, so if this one, you presumably start from blue, something that's blue to something that's pretty yellow or clear, right? And then you have the absorption of this one. I think a lot of the patterns look, when you did it like this, it looks like blue is the first one is usually the most blue, right? And then they kind of went like this, and then they went up when they got to yellow. They kind of did something like this or something, but yeah, in any case, you could do it either way, but I do want a graph of the values, right? Do you guys all know how to do that on Excel or on sheets or whatever? Okay. So if you didn't do a graph, then please reshare it, please correct it and then reshare it with me, and then okay? We're good? Were the labs okay? Did you guys understand what happened in the labs? Yeah. The questions in the labs are okay too, so you'd be able to answer them? If I asked you, if I gave you a different scenario and I said there are 2 colors, this one eluted faster, then you would know what about those molecules, that those molecules are what compared to the ones that those are larger, right? Whereas in gel electrophoresis, though, I have a band pattern that I'm giving you, right? And suppose if there's 1 band, that means you know that how many different sized molecules are in there. Just 1. If I have 3 bands, how many different sized molecules are in there? 3. If I have a band that goes to toward the positive one and towards the negative one, right? Then you'll know that one positive and 1 negative, and you know that there are 2 different sized molecules, different types of molecules there, right? And you'd be able to know that the one that travels farther is the shorter one in this case, right? So it's kind of opposite of the gel column, right? We're good? All right, today we're starting lipids. This is our next biomolecule, so this is exciting. All right, you guys have graduated from, yeah. Yeah.
About the gel column, so when you, I just forgot, I'll ask you.
Okay. All right. Closing this door. Yes.
Are we getting our quizzes back today?
Yes, I'll give you your quizzes back today if we have enough time. Did you see I posted them in classroom? Right? Were you happy with your quizzes or not happy with it? All right, so some of you guys, it's sort of like the time. We've had a few major quizzes now, and so I need to put you on some PIPs, okay? So I didn't want to do it, but I think it's good for you, and, if you can get off the PIP if you change if you kind of reverse your performance. So, all right, I'll be in touch about that then. Okay, so today we're going to start lipids. What are these pictures of, do you think? Lipids? That would make sense, right? All right, so this is what? Butter. This is honey's not. So the thing is supposed to be wax. It's supposed to be kind of like beeswax, right? And then
That's
this,
honey.
not honey. Oil, right? It's like some sort of like vegetable oil or. All right, not honey. Does that look? I guess it kind of looks like honey. Slightly ambiguous. I apologize. Okay. So today, what we're going to do first, we're going to do pretty much how we approach any new biomolecule. We're going to define the characteristics and the functions of them, of lipids. You guys might know a few of those, so you can help me. I'm going to introduce all the five or six different classes of lipids, and we're only going to do the first class today. Tomorrow, we're going to do the rest. Right? I hope. Then we're going to distinguish between something that's called saturated and unsaturated, right? You guys know, you've heard of that, right? Yeah. Do you know what it means? No. It has to do with food, like fat, unsaturated or saturated fats. But I'll show you the actual structure, like what that means structurally. And then you guys are going to build your models again, right? So you guys are really familiar with those things, right? Those kits. So I think it will be easier this time around because you guys are already familiar with them. Okay. So the first thing about lipids, right? What do you know about oil, or what do you know about wax, or what do you know about butter and its relationship to water?
Oil's hydrophobic.
Exactly. Good. All of them are hydrophobic, meaning they don't like water. So what kind of bonds do you think they have within their structure? Neutral. What are they called? Huh? Non-polar, right? So they consist mainly of non-polar bonds. Can someone give me an example of a non-polar bond? What's the main two things in organic molecules that George?
Like a carbon bonds to another carbon.
Perfect. Carbon and carbon. So it's going to have a lot of carbon-carbon bonds, right? Because neither of them can pull the electrons from each other because they're equal, so it's just non-polar, right? What else does carbon like to bond to that it doesn't? That's kind of like an equal tug of war there too. Hydrogen, right? So a lot of times in lipids, you're going to see you can expect to see a lot of non-polar bonds, so CHs, a lot of CHs. What about a polar bond? What's an example of a polar bond? Omar? Oxygen and carbon, right? So if you see oxygen and carbon, that means that it's polar, so that means the oxygen is going to be hogging the electrons, right? So that would make it polar, right? Do you think we're going to see a lot of those? No. Might we see some of them? We might see one in each fatty acid, but we're not going to see a lot of them, right? Okay. All right, so if we're saying that they're hydrophobic, would they be soluble or insoluble in water? Insoluble. They don't even want to be anywhere close to water. So they're insoluble in water or in polar solvents. You guys remember what a solvent is, right? Okay. So it's insoluble in water or polar solvents. That means what would it be in non-polar solvents? Then it would be soluble in non-polar solvents. So they do like things that are not charged. They don't like things that are charged. Lipids for the most part, right? And then, like I said before, you can expect long chains of straight up carbon, hydrogen, carbon, long hydrocarbon chains. It's another name for them, hydrocarbon chain. Okay? Any questions so far? Oh, I forgot to give you guys, did I give you the packets or no? No. So the packet, it's not in order necessarily, it doesn't go actually, logically through the, so you might have to skip around. So I recommend maybe doing this one at the end, afterwards, but I'll pass it out now, just for those of you who want to get a head start on it. Can you ask it after? Yeah, just write it down so we know what it is, but it's not I want to sort of stick with this right for now. All right. All right. So let me talk to me. Yes. I don't have it. All right. So functions of lipids. So think about like lipids in your body, right? What do we have a lot of, like, well, maybe not a lot of you guys do, but like people have a lot of fat, right? Why do we have fat? Okay, so it could be for insulation. It's energy storage, right? That's the main thing. That's the main thing that fat does, right? So, and because it has so many more just carbon hydrogen bonds, it has these long chains. It's way better at storing. It's almost 2 times better at storing energy than carbohydrates are, right? So it's really good for energy storage. The next function that it has is our cell membranes, right? And if you look at these, there are actually 3 different molecules that you might be able to notice here, right? You see that thing right there? Yeah, it's like a 4-ringed thing. That's a lipid. This little guy with the little tail and the head, that's a lipid. And then this one over here, you guys know what this is already, right? Well, so this is an oligosaccharide here. That's a glycolipid, right? So all 3 of those things are members of the membrane, right? So they're going to hold important functions for stability, structure, and signaling, right? Speaking of signaling, we have all these other 4-membered molecules, like rings, right? 4 fused ring molecules, right? That are involved in signaling. So like hormones. Have you guys heard of testosterone, estrogen, progesterone, aldosterone, right? Those are all lipids, right? They're these specific types of lipids that have 4 rings, right? And then finally, you guys said insulation, right? But so this is just an example of a plant, right? On the outside, you know, it has like a waxy sort of coating. That's called the cuticle, and that can be used for protection, because when water falls on that, what happens? It slides off because that stuff is hydrophobic, right? Insulation. So if you have like a lot of extra fat stores, you can it can cushion other like vital organs and things like that. And then for padding, padding, and then insulation to keep you warm, right? Because it keeps heat in. Okay? Can I go on? All right. So let me see how many there are. 1, 2, 3, 4. I guess we're going to talk about 5 different classes of lipids, right? The first one we're going to do is fats or triglycerides, and that's the one that we're going to talk about like mainly today, and you guys are going to build the models of them. I'll erase that. Okay. The next thing is phospholipids, right? And that was one of the ones in the membrane that you saw. So that's involved in like membrane composition, right? You guys know what glycolipids are already, so those are part of the membrane. And what was their main function? Remember the glyco portion is what type of carbohydrate? What level of carbohydrate, Hannah? How many little monomers are there on those usually? Between like, no, I thought you were like starting to answer, so I thought you knew, but Jasmine? 3 to 10, right? So that means those are oligosaccharides. What was the role of oligosaccharides again?
To like signal into
Recognition and signaling. Good. So that's what glycolipids are going to be mainly for, but they're in the membrane, right? Steroids. So steroids are for signaling, and those are like those 4-membered things. All those things with the 4 members, those are all steroids. And steroids are lipids. And then the last one we'll talk about is lipoproteins, and we'll do that in like the next, like I think either tomorrow or Monday. So I think we're going to have our quiz on this maybe on Monday or Tuesday. Is that okay? Okay, because I don't know, somehow we got like a little backed up with those labs and stuff. Okay, so this is just like overview, and we're going to tackle each one at a time. Good? All right, so functional groups that we know. What do we know from our carbohydrates? We know 2 functional groups. And some of you guys were a little confused on this, on the quiz too. So what I wanted to see was, Addie, what's one?
Hydroxyl and
Good. So I wanted to see hydroxyl, which should look like this, right? If you drew it like this, or like this, or like that, I gave you credit, right? And a carbonyl, which is what? C double bonded to an O. Some of you guys drew it like this, but what I really wanted to see was what? I just need to see those extra two things because I need to know that carbon's bonded to two other things too. But really, this is the carbonyl group, right? So we have two groups. We have carbonyls and we have hydroxyl. Hydroxyl and carbonyl, right? You guys are comfortable with those now? And if I asked you to identify the functional groups of carbohydrates, you'd be able to do that, right? All right. This is on like one of the early slides in carbohydrates, but yet like a lot of people, instead of writing these as the functional groups, you guys wrote like aldose and ketose, which like those are both they're types of carbonyls, but they're not actually like the functional groups of carbohydrates. That's like these are the ones that are considered that, right? Lita?
So functional groups are static for all molecules.
So basically,
if I see a hydroxyl group anywhere, it's hydroxyl.
Yes.
Okay.
Yeah. And when you see a carbonyl too, like usually functional groups don't really contain C's and H's because those are like so abundant. But when you see something like a C bonded to an O, or if you see an OH hanging off of there somewhere, then you know that's like a functional group. It's like a marker, right? All right, so we're gonna learn a new one, and the new one is called carboxyl. And so, what can you sort of see in both of, like what can you see in that name? You can kind of see George.
Carbon and oxygen.
Carbon and oxygen, right? So what were the two groups that we knew before? Carbonyl, right? And then hydroxyl. So this is kind of like a mix of both of those. So if you were to mix both of those together, you'd get, carboxyl, right? Do you see what we did? So instead of an R group on one of them, it's just a hydroxyl group attached to the carbonyl carbon, right? And if you see how it's written, there's like there's one carbon, two oxygens, and one hydrogen. So sometimes it's abbreviated like COOH, right? Just like that. So I'll have like a molecule. Maybe I'll do like some molecule like this, and at the end I'll have like do something like that. And that just means that this is basically C double bonded like that, right? And anything that contains this, so if you see this like group on any molecule, that whole molecule is called a carboxylic acid. So this, once you see that, it's a carboxylic acid because it has a carboxyl group. Easy? All right. So now we're going to talk about, so what was the monomer of carbohydrates? Do you guys remember? Monomer of carbohydrates. Monosaccharides, right? And so the monomer of lipid is called a fatty acid. It's not the same way though. We're not combining fatty acids next to each other. We're actually combining them with a third molecule, or like a separate molecule. But in any case, you need to know. Like when I ask you what the monomer of a lipid is, you'll say just like fatty acid. That's what we're considering it to be, right? So a fatty acid, basically, it's composed of a carboxyl group and then a hydrocarbon tail, right? So like, here's my hydrocarbon tail, right? Why is that a hydrocarbon tail? Because it's composed of hydrogens and carbons, right? And then it has the carboxyl group here, right? So this whole thing together is called a what? A fatty acid, right? What do you guys notice about the ratio of carbons to hydrogens to oxygens? Basically, the main thing I want to see is the ratio of carbons to oxygens. Lita?
It's basically almost a 1-to-1.
Carbons to oxygens?
Wait, no.
Sarah? What's the ratio of carbons to oxygens?
None to.
How many oxygens do we have?
Oh, to oxygens too.
Just two, right?
Yeah.
So basically, like the idea is, like, and what was the ratio in carbohydrates between oxygens and carbons? It was pretty much 1-to-1, right? Is this a 1-to-1 ratio? No, it's basically a ton, and we could have this go on for like ever. The carbons could be like, it'd be like not infinite, but a lot of carbons, but this part is always just 2 oxygens, right? So the ratio, you can usually identify a fatty acid by noticing that there are only 2 oxygens and there are a ton of carbons, right? So if I gave you like a formula where it was like C like 25, H, and then usually it's like about double, so like H 50, 50, and then O 2. What could you immediately probably tell me that this is? Fatty acid, right? Would you say it's a carbohydrate? No, because if it were a carbohydrate, this would have to be what? 25, right? So just notice immediately like the way lack of oxygens in fatty acids or in lipids, right? Okay. This end right here, so the non-carboxylic acid, like the carboxyl group end, is called the omega end. And we'll talk about that in a second, what that means, but that's really important to know. Okay? And so this...
Carboxyl side?
The non-carboxyl side, yeah. So the opposite of carbon. And then this fatty acid is considered the monomer of lipids. Bless you. Are we good? Okay. Okay. All right. 2 types of fatty acids. Saturated and unsaturated. How are you going to tell the difference? What is something that means if it's saturated? Suppose I have a sponge and it's like completely saturated.
It's really full.
So that means every possible hole that is there is completely full, right? All right, so in this case, too, it means kind of the same thing when we're talking about carbons in a fatty acid chain. So in a fatty acid chain, for every carbon, it has as many hydrogens as it could possibly have on it, right? So it's like it has the maximum number of hydrogens that it can have. So there are no double bonds in this, right? It has the maximum number of hydrogens, right? Whereas in this one, what do you notice here? So instead of having CH2, CH2, CH2 for all of the C's that are there, how many C's does this one have? Just how many extra C's does this one have? How many extra H's does this one have right here? Just 1. And how many extra H's does this one have? Just 1. And instead, they made a double bond with each other, right? So that is considered unsaturated. Okay? So an unsaturated fatty acid is one that has either 1 or multiple double bonds. And we're not talking about the double bond here between carbon and oxygen because all of them have that, right? We're talking about carbon-carbon double bonds. Okay? So if it has 1 carbon-carbon double bond, what do you think it's called? What's the prefix for 1? Mono. So if it has 1 carbon-carbon double bond, it's called a mono unsaturated fatty acid. That makes sense, right? Because it's only unsaturated by 1 double bond between carbons, right? What do you think if there are multiple? So suppose I wanted to continue this down, and instead I made this, right? Like that, right? So here's another double bond. See that here? So now it's not a mono unsaturated saturated fatty acid. It's called what now, George?
Poly.
Good. Now it's called a polyunsaturated fatty acid. If there's if there's more than 1 double bond. Okay? All right. What's the shape of this one, the unsaturated one? What shape does it take? We do?
It's pretty straight.
Straight, right? What about that one, the unsaturated one? What shape is that? Anthony?
Like branch.
It's not exactly branched because branched would be like that it would have like an equal thing that comes off of here, but you're close. It's called bent. They're just called bent. Okay? So unsaturated ones are bent. Saturated ones are straight, right? If this is straight here, and I want to pack a lot of them together, right? Can I pack a lot of them really closely together so they become like really dense? Yeah, right? So then do you think this is going to be solid or liquid at room temperature? Solid, because I'm like packing them really tight together, right? So that's solid at room temperature, like for example, butter, right? Butter would be would be a saturated, like because it's totally has as many hydrogens as it possibly can, right? What about this one? If I have all these like bent fatty acids and I try to really like cram them together into like this dense configuration, do you think it's going to work? No, because they have that like weird kink in them, right? So those are usually liquid at room temperature and those are like in the form of oils. Okay? Where do you think most of this kind of solid stuff comes from? Like plants or animals? Usually these saturated ones are derived from animals, right? And then usually the liquid ones are derived from plants, right? There is one major exception. Do you guys know? So usually when you have butter or like lard, you're getting that from animals, right? What kind of oils do you usually have? Do you ever say like, oh, I'm using like pig oil in my on my salad? Vegetable oil, corn oil, olive oil, but the exception is fish oil, right? Because fish produce these omega-3 fatty acids that are really good for us, but those are liquids, right? So that's the one exception. That's good. A major exception. Okay? Can I go on? All right, so those are animals, those are plants. Bent, straight, one or more double bonds, no double bonds, right? Okay. So let's look at the ones that do have the double bonds, okay? So those are called what again? So those are called unsaturated. And I want you guys to look at the difference between this molecule and this molecule. It's the same number of carbons, right? I made them the same, and the double bond is in the same position. But what's the difference between this one and this one? Can anyone sort of get that, Mike?
One's bent and one's straight.
Okay, one's bent and one's straight. But look at the locations of the hydrogens.
One the H is up, and on the second half. Oh wait, after the double carbon bond, one of the H's is up, and then the first H is down.
Good. So on this one, they're both on the same side, right? And on this one, they're on opposite sides, right? Okay. So the one when they're on the same side, right? It's called cis, right? The relation. So cis or trans can only refer to a double bond, right? And they refer to whether these hydrogens are on the same side, right? Like they're both coming off of this side, or whether they're across from each other, meaning they're on the opposite sides of it, right? So what shape is a cis bond? If the hydrogens are on the same side, then what shape is it going to be, George?
Bent.
It's going to be bent, right? And if it's bent, what kind of like state will it be at room temperature? It will be liquid, right? Because they can't stack together very tightly, right? So most naturally occurring unsaturated fats, they're naturally cis, right? This is a good thing to have cis bonds because then they're liquid and they're natural, right? So like vegetable oil, olive oil, like all those oils, they're naturally liquid, they're naturally bent, right? Cis, right? Look at the trans one though. So trans is because the H's are on opposite sides or they're across from each other, right? What shape is that though? Because they balance each other out. See like how they're kind of like balancing? It becomes actually straight, right? And so is that a good thing or a bad thing? If it's straight, then what can you do to them at room temperature? You can really pack them together so it's solid at room temperature, right? And actually, trans fats do not normally exist in nature in mammals, right? This isn't a good thing. The way this happens is by this artificial process called hydrogenation, which basically it adds hydrogens to double bonds and then it gets them to sort of flip from their normal state to this state. So then they become solid, right? And do you guys know like Crisco and like those types of like sort of like artificial like shortening, like they're like solid even though they were originally like oils, right? Those like that's like it's like originally supposed to be an oil but it ends up becoming a solid, and then it's like those are really not healthy for us, right? So you have to know the difference between a cis bond and a trans bond, right? If I gave you a picture of those, right? And you have to know the shape. Good? So a trans bond, a trans like unsaturated fatty acid is pretty much the same effect as what? If it's straight, it's pretty much the same thing as a saturated fatty acid, right? Because they're straight and they can stack together and be solid. Okay. Good? Not good.
No, like I'm still copying.
You're still copying? Okay. make sure you guys can. Okay, we're good? Yeah?
What was the name for fatty acids with carboxyl?
Say it again?
What was the name for the fatty acids with carboxyl?
Is that a question on the thing or
No, I just didn't catch the name.
the name of?
Like when it
Oh,
has
oh,
carboxyl
oh.
in it.
Oh, it's called a carboxylic acid. Carboxylic. Sorry, I wasn't sure what the question was. Okay. Yeah, carboxylic acid. Okay. Good? Okay. So now we're going to make fatty acids are the monomers. So now we're going to make a triglyceride. Bless you. So for a triglyceride, the recipe is essentially this thing. You have to memorize what this is, right? Is this a fatty acid?
No,
Why?
it doesn't have a carboxyl.
Doesn't have a carboxyl group, and what does it have?
It has hydrogen.
It has hydroxyl, right? Which clearly makes it hydrophilic, right? So this is not a lipid, but this actually goes into making a triglyceride. So we need to memorize what this is. It's basically 3 carbons and then 3 hydroxyl groups coming off of one end, and the rest are all hydrogens, right? So I ask you to draw glycerol or make a model of glycerol, you would draw that, right? It's pretty easy to memorize, don't you think? Yeah? Okay. So to make a triglyceride, you need 1 molecule of glycerol, right? And then you need 3 fatty acids. Oops. All right. Do you know why are these all fatty acids? What makes this a fatty acid? It has George,
It has the carboxyl group
carboxyl group, and then
and the hydrocarbon chain.
hydrocarbon chain, right? What about this one? Same.
Yeah.
But they're all different length fatty acids. You guys recognize that, right? But it doesn't matter as long as they're all fatty acids and they have that carboxyl group at the top. Then we can add it to glycerol, right, Mike?
Could they be the same length, or do they?
They could all be the same length. We could have one in here that has like a double bond and that's unsaturated. It doesn't matter as long as they have this part. That's the part that's going to be doing the actual interacting. But yeah, they could all be the same. They could all be different. They, whatever. They could all be bent, unsaturated, anything, right? So those are the 3 fatty acids, and there's glycerol, right? Can anyone sort of see a way that they could possibly? Can anyone predict how we're going to get these 2 molecules, or these 4 molecules, I guess, together into one? I'll give you a hint. How am I going to bond these together? Mike,
You're gonna bond the OHs to carbon with oxygen.
Wait, what did
What?
you just say?
What OHs? These OHs or these OHs?
Those OHs, the one that you're figuring out. You're gonna bond those to carbon with oxygen.
Bond these to carbon. I don't really understand what you're saying. Maybe you're thinking of the right way.
So like you take the OHs on the bottom of the glycerol
Okay, glycerol.
and bond them to carbon.
Okay.
So and then just like.
By what process?
Wait, it'd be a something linkage.
Close. You're kind of, Kaylin? By dehydration synthesis. The process is dehydration synthesis, right? And then the thing that I'm making is going to be similar. So the process is the dehydration synthesis or condensation, right? But the process, the thing that I'm making would be similar to glycosidic linkage, but it actually has a different name. So let me just zoom in a little bit to make it a little easier for us to see, right? So what do you guys, what 2 things are going to interact in dehydration synthesis? I just chose those just because they were easier to kind of get rid of for my model here. So do you see how here's an OH and here's an H, right? Here's an OH, here's an H, here's an OH, and here's an H, right? And so what is that going to make? So I'm going to get how many molecules of water coming out for when I make 1 triglyceride? 3 different molecules of water, right? So this is dehydration synthesis. You guys knew that. So I'm going to take the water out, right? So the water's gone, right? So now what do these want to do? Does this want to have only 3 bonds? No. It wants to connect to the oxygen, so it will do so, right? And then this bond, like Mike said, it kind of looks a little bit like what other bond that we know of? It looks like a glycosidic linkage, right? But in this case, because it's a fat, this is called an ester linkage, not a glycosidic linkage. So if I ask you what the bonds in lipids or in triglycerides are, you would say ester linkages. When I ask you what the bonds are that connect carbohydrates, monomers, right, you would say glycosidic linkages, okay? So you see like how they're analogous to each other? And you see how this whole process works, right? You guys, this is like nothing surprising, right? Nothing special? Okay. All right. Here I want to talk. So remember we mentioned omega, right? Which is the omega end of a fatty acid? It's the one that
The one that doesn't have the carboxyl.
Good. It's the group. It's the one that doesn't have the carboxyl group on it, right? So it's like the far end, right? Have you guys heard of omega-3 fatty acids? Yeah. Or omega-6? Yeah. Those are certain like oils, like fish oils, right? That we can't produce, but we can take supplements of them, right? And they're really good for our brain and like for our bodies, right? So the way that they call it an omega fatty acid is you take the carbon that's the furthest away. So and you name that number 1, and then you count backwards until you reach the first carbon of the double bond, right? So here's my first. So I start here and I go 1, 2, 3. So then this is called a what? Omega-3 fatty acid, right? What about this one? I start at this carbon. I start at the one all the way at the end, and I count back until I reach the first one that starts the double bond. So 1, 2, 3, 4, 5, 6. So this is a omega-6 fatty acid, right? We're good? You guys are great. All right. I just want to go through one. So what is this? I want like 2 different descriptions to describe this, right? Think about it for a second. 2 different descriptions. I guess 3 different descriptions. 3 levels, 4 levels of description. So think about it. I want all 4 things. Okay. Charlie, what's one?
Carboxyl.
Okay, well this is the carboxyl group, but what is this whole thing? I want like the description of the whole thing. It's a, what'd you say? Yeah. Did you say something or no? No. So you can call this a carboxylic acid, right? All right, so it's a carboxylic acid. What else?
Unsaturated.
Is it saturated?
Unsaturated.
Did you say unsaturated or saturated? I heard saturated. Unsaturated. Okay.
Yeah.
Sorry, we're having like communication breakdown today. Unsaturated. Yes. Okay. Sorry, I only heard the saturated part. So okay. It's unsaturated. Unsaturated what?
Cis.
It's a cis, and then
Monounsaturated.
okay, monounsaturated. George.
It's omega-3 fatty acid.
Omega-3, and so it's a carboxylic acid, but it's also a fatty acid. I wanted that too, fatty acid. Right, so you have all these different ways of describing it. Carboxylic acid because it has a carboxyl group, right? Fatty acid because it has a carboxyl group plus a hydrocarbon chain. Monounsaturated because it has 1 double bond. Cis because both of the hydrogens are on the same side of the molecule. Omega-3 because we're counting backwards from here, so we do 1, 2, 3, right? Is that, everyone got those, right? All right, so what I want you to do though, do you remember what I said about notation, right? Sometimes when we were making glucose, right? Remember, instead of drawing it out all the way like this, right? Right, instead of doing all this, right? Remember they just started doing it like this. And you still have to do the other things that aren't carbon, but then they would just do like remember they would just do things like that, and then like so any point here represented a carbon, right? Do you guys remember that? Did I told you that? Alright, so if I want to do that here, I want to remove any like C's and H's, right, from this and just make like a line for where the bonds are. What I'm going to do is I have to keep this the way it is, right? because these are not carbon or hydrogen, right? So I'm going to get rid of this carbon. I'm going to get rid of this carbon and these H's. And instead, I'm just going to put a line between those, right? That shows that this is a carbon and this is a carbon, right? And for this one too, again, I'm not going to put any of these here. I'm just going to put another line for that carbon, right? So basically, it's going to look like something like this, right? Can you guys see how this one came from that? All right? So maybe it's easier to fill it in here. So this right here, this point, was this, right? It's that carbon here. This next point is what? It's a carbon and it has whatever H's, but the H's we're never going to draw in anymore, right? And then this one here is the next carbon, right? So that's this one. This one's this one, right? This one is the first one of the double bond over here. And then here's the other carbon of that double bond, right? The hydrogens are on the top part, so that means that this is cis, right? And then here's another carbon here. And don't forget that this thing right here is another carbon too. Right?
So how do you know what direction the line is going?
Which direction the
Like,
lines?
why is the first one going down?
It doesn't matter. It could go up. Like, so I could have done this, and I could do it the other way too. Right? Good question. All right. So do you guys sort of understand how this notation works, George?
So like the second line, like that extra line underneath, like the RB, like carbon chain, like to the right that shows like the cis.
This one?
That's just so that's cis, and it was a straight. What would it be?
If it were straight, then it would. If it were trans, then this would just continue up like that.
And that wouldn't be like that second line underneath it.
But then it would be like totally. It would just look like there would be the double line because that's the double bond between the carbons. But it would just look like that because it means that it's straight, and one of the hydrogens would be like presumably over here, and the other hydrogen would be like over here, right? All right. So you guys can see how that works. Don't forget. So if I'm going to say what kind of omega fatty acid is this, which carbon do you start from? Right here, right? So remember this thing right here, that's the first carbon, right? I'm going to give you some practice ones to do. Right? Okay? This is fun, right? So this is how you're going to see it when you take orgo. Right? This is what it is. Organic chemistry? It's like the most, like, a lot of people like dread that class, you know, for pre-med, when you're taking like pre-med stuff, that's like, it's like a language though, it's kind of interesting. So if you like that kind of stuff, then it's interesting. But, alright, so tell me about this one. What is this? This is what? This is a fatty acid, right? Because we have a carboxyl and then a hydrocarbon chain, right?
Omega-3.
So it's this one?
Yeah.
Alright, so where are the carbons?
Oh, omega-6.
Right? Okay. So this one's omega-6 because it goes... So remember to start all the way from that point right there, right? All right. What about this one, Mike? Where do I start?
There.
Okay, 1, 2. So it's an omega-3. And is this monounsaturated or polyunsaturated?
Poly.
Is this one monounsaturated or polyunsaturated? Poly, right? Okay. Let's see. Can you guys see that or no?
Yes.
You see it now? All right. So, someone tell me what this one is. Where do I start counting? I start here, so 1. Okay. So omega-9, monounsaturated fatty acid. Are we good? And look at, do you guys notice, can you appreciate how many carbons and hydrogens there are in this part? So is this going to like water or not? It's going to not like water, right? This part of it is going to like water or not like water? That part actually will like water, but because this is overwhelmingly like a larger part of the molecule, lipids in general, we're saying they're hydrophobic, right?
See, you have omega
omega anything really, as long as you're counting them back from. Right? So make sure you know which carbon to start from, right? Okay, so your task, what do you think you guys are going to do today? So we're starting with, so each group is going to, you're going to get 4 of the kits, but you don't all have to build a triglyceride. Your group collectively has to build a triglyceride, right? So, but the thing is, you can't just build a fatty acid with like 3 carbons. That's like not, that's why. Right? So minimum of 6 carbons in the fatty acid chain, right? You can do more, you can do less. You can make it monounsaturated, you can make it polyunsaturated, but you do have to do the ones with like, huh? You guys, that's a good question. You guys have to figure that out. So how you have to make a carboxylic acid group, right? And that requires a double bond between carbon and oxygen. Figure out a way to get these to make a double bond between carbon and oxygen, right? I don't know if there's enough here, but if you guys are missing some, you can kind of like reduce it, okay?
Here.
You guys all have one, no? Alright, maybe you guys should go to a lab table. Dynamic issues here, how is so that should unsaturated fatty acids. 1 omega-3 unsaturated cis, 1 omega-6 unsaturated cis.
Alright, you're getting the 1 omega-3 saturated, unsaturated cis, and then you can do the 1 omega-6
And you guys, for the omega-6, I don't want like that, the sixth, the other carbon to be the one of the carboxylic acid either. I want you to add like 2 more beyond the double bond so that before it gets to the carboxylic acid. Right? So that will be like maybe like a 9 carbon. Do you understand what I'm saying? Before you make the triglyceride, I want to see what you have. Right, Juliana, can you guys come up here when I'm going to give you your quizzes back. So you can look at it real quick over here.
Yeah. I have to do it by hand.
Why don't you look through those and tell me and like see if you can find the correct answer here. Let me give you another pen. All right, Anthony. Just take a look, and I want you to tell me what the actual answers are supposed to be, and then before you go back to your seat, all right? Let me give you a pen. Yeah, will you circle the correct answers for the ones you got wrong, right? Charlie, come on over and take a look. Did you get them, or did you figure them out, or no? Mike? You are so easy, but you're not going to actually get a pip yet because I don't think you know how to fix the problem. Yeah?
I studied for it.
Alright, so I want you to look through here for 5 seconds. Like just sit here real quick, and then I want you to bring in the food and alright, and figure out the correct answer. Okay? Yeah. You guys, actually I want you guys all to do this right now. I want you to go, I'm going to give you your test back. For the questions you got wrong, listen please. I don't want you to be like to the person next to you, what was number something something. Like that's, if I hear that, then you're getting a 0 for the whole thing. But you can't do that. I want it to be totally silent. You have to figure it out on your own. Like don't rely on someone else to like, you know, this should be something, this is your, you guys have to do it yourself, okay? Don't ask anyone else. So when you get your test back, in another pen color, I want you to circle the correct answers for the ones you got wrong. Alright? So I want everyone to kind of pause what you're doing right now and then just do that real quick for me. And then I'll collect them back. Alright? So everyone should be please write on them and circle the correct answer for the ones that you got wrong. If you've gotten them wrong, then just review it and see what you got right. Alright? But everyone should, and it's funny because in every class, there's usually someone who gets a perfect score on almost every class. There's usually like one, so far in my experience, there's been like one person per class who's like the,
Jen.
I'm not saying or confirming anything, but there are, in every class that I've had, so I've had like 4, right? There's always been like one who. It's funny because there aren't 2 usually. There's always just one, and. Huh?
Wait, you said there are 2.
Yeah, I mean, so far there haven't been. There's always one person who gets. But then it's funny because like everyone's grades are also like usually in like a certain order. Like even, I know, like there are some people who always like get 1 or 2 wrong, some people who get 3 wrong, and it's usually like very consistent. You'd be surprised at the sort of statistics, like the way it's very interesting. person, right? But then I was like, oh wait, I forgot to grade this whole page. So that's why your score probably has something out of 69 and then it adds 6 or something out of 6 to make out of 75, right? Do you understand which ones you got wrong?
Yeah, most of them are too.
Did you circle the correct ones? Yeah. I think so. I hope so. Have you guys circled your correct ones? What? No, but she can't. That's what she's going to figure out. You can't. Yeah.
Oh, it's just here.
Yeah.
It ends
No, that's. Yeah. Finish? Yeah. Sarah and Mike, I need you guys to come back here and start together.
Wait, no. No, yeah.
I need those carboxyl.
I'm sorry, guys. I think it's my time.
If you take your test back, oh, don't forget there's someone's phone here. I need to have all your tests. Do I have them? Finish the worksheet. 1, 2, 3, 4, 5. Thank you. 6.